Welcome back, everyone. I am absolutely thrilled you're here today because we're tackling something that is literally the blueprint of life. Welcome to today's Explainer, where we're going to decode that blueprint by diving into genome annotation. We're going to make sense of the incredibly complex genetic data that makes us, well, us. So let's just jump right in.
There's this massive misconception out there. People think, hey, we sequenced a genome. Now what? We're done, right? We hear the big news announcements, the confetti drops, and we kind of assume crossing that sequencing finish line means the job is over. But actually, it's just the starting line. Sequencing is really just gathering the raw data. The real magic, figuring out how an organism actually operates, comes next.
Think about it like a giant instruction manual. Raw, sequenced DNA is basically a massive, unformatted text document. I mean, for humans, we're talking about 3 billion letters with absolutely zero spaces, no paragraphs, no punctuation, just a wall of A's, C's, T's, and G's. Without a translator, it's completely unreadable. Genome annotation is that vital translator. It's the engine that takes this overwhelming block of code and turns it into a highly organized, searchable manual. Okay, so here's our roadmap for today's explainer. We're going to cover 1. Decoding the DNA blueprint, 2. Structural versus functional annotation, 3. The annotation workflow, 4. Gene prediction and databases, and finally, 5. Real-world impact and the future. Let's get to it. Kicking things off with Section 1, Decoding the DNA blueprint. So officially, genome annotation is the process of identifying genomic features, like genes and regulatory elements, and assigning functional information to them based on solid biological evidence. Basically, through a mix of computational and experimental methods, it acts as the ultimate biological translator. It single-handedly transforms raw sequencing data into actual, meaningful biological information. Literally, without this step, bioinformatics would just grind to a halt. And this brings us to the crucial so what? Why do we pour so much computational power into this? Well, the impact spans across some pretty massive fields. By identifying protein-coding genes and predicting what they do, we're able to support critical disease research and speed up targeted drug discovery. It lets us do comparative genomics to understand evolutionary biology, and it directly improves crop and livestock breeding. It's no exaggeration to say this process is the absolute engine driving modern biological applications. Moving right along to Section 2, Structural versus Functional Annotation. To really grasp this, you have to look at its two main pillars. First, structural annotation. This is all about identifying the physical components and their exact coordinates. Where does the gene start? Where does it stop? Where are the exons, the introns, the promoters? It's kind of like mapping out the rooms and hallways of a giant building. Then you have functional annotation, which predicts the biological roles. What does this newly discovered protein actually do? What metabolic pathways is it part of? Where does it live in the cell? If structural annotation maps the rooms, functional annotation figures out who works in those rooms and what their specific jobs are. You absolutely need both to understand the whole shebang. Let's look at how this is actually done in Section 3, the annotation workflow. Think of this as the bioinformatics factory floor. Step 1 kicks things off by sequencing the genome and assembling those tiny, short DNA reads into longer, continuous sequences. Step 2 acts as our filter, identifying and masking repetitive DNA elements so they don't completely confuse our algorithms later on. Then Step 3 is where the heavy computational lifting happens, predicting potential genes. Steps 4 and 5 are the structural mapping and functional assigning we just talked about. And finally, Step 6 is validation. This is absolutely critical. It's where all those predicted annotations are rigorously tested against real-world lab experiments, transcriptomics, our existing published evidence to make sure we didn't just invent a phantom gene. Which brings us to Section 4, gene prediction and databases. When researchers are actually predicting these genes, they're constantly weighing trade-offs between three main methods. First up is ab initio prediction. This relies entirely on sequence patterns. It's incredibly cool because it doesn't need a reference genome at all, making it a game changer for newly discovered organisms. But yeah, it definitely struggles with accuracy in really complex genomes. Then there's homology-based prediction, which compares sequences to genes we already know. This gives you super high accuracy, but you're totally dependent on actually having a reference available. Finally, RNA-seq assisted uses actual transcriptome data. This one is phenomenal for detecting things like alternative splicing and definitively proving that a gene is actually being expressed.
Okay, so once we predict the genes, we have to figure out what they actually do. That's where functional annotation databases come in. You can think of these as the massive reference dictionaries we need for our translation process. We've got the gene ontology, or GO, which classifies biological processes and cellular components. KEGG is our absolute go-to for metabolic and disease pathways. UniProt is the comprehensive library for protein sequences. PFAM identifies conserved domains, and InterPro essentially integrates signatures from multiple databases to give researchers a really comprehensive, fully fleshed out protein annotation. Now, to query all those massive databases, researchers rely on a highly specialized arsenal of bioinformatics tools. You've probably heard of BLAST. It's universally famous for comparing sequences to identify homologous genes. PROCA is this incredible automated tool designed specifically for bacterial genomes, while Augustus is the standard go-to for complex eukaryotic genomes. Maker is an integrated pipeline that pulls in multiple evidence sources to really maximize accuracy. And RAST is widely used for the rapid annotation of both bacterial and archaeal genomes. So, different tools for different jobs, right?
And finally, let's zoom back out for Section 5, Real-World Impact and Future. All of this microscopic, highly computational work, it is the bedrock of our modern world. In medicine, annotation is literally what allows us to pinpoint disease-causing genes. It's how we map out targeted cancer therapies and unlock personalized medicine. In agriculture, it's the secret to engineering resilient crops and breeding disease resistance to secure our global food supply. In biotech, it drives synthetic biology. And over in microbiology, it is our absolute first line of defense for detecting antibiotic resistance and characterizing dangerous new pathogens before they have the chance to spread. The really great news is that this once tedious pipeline is undergoing an absolute revolution. Modern innovations are dramatically enhancing the speed, accuracy, and completeness of genome annotation. We're seeing artificial intelligence and machine learning automate some incredibly complex pattern recognition. Long-read sequencing technologies are finally helping us navigate those highly repetitive, messy DNA sequences. Plus, when you add in single-cell genomics, multi-omics integration, and massive cloud-based platforms, we are now annotating complex genomes faster and more accurately than at any point in human history. With this rapid evolution of computational methods, we are hurtling toward a future of precision medicine and continuous genomic learning. So I'll leave you with this. How will AI-driven, real-time, personalized genome annotation fundamentally change our healthcare? Imagine a world where your individual genome is perfectly, functionally annotated in real time to predict and prevent illness before it ever happens. The blueprint is there. We are just getting better and better at reading it. Thank you so much for joining me today on this explainer. Keep learning, keep questioning, and I'll catch you in the next one.